This is the second lecture on proteins. So let's continue from where we left off in the previous lecture. The previous lecture was talking about disulfide bonds and how they stabilize the shape of a protein. Most proteins in the cell, and outside the cell for that matter, do their job by binding to something. Whatever a protein binds, whether it's a carbohydrate, another protein, a mineral, a vitamin, that object is referred to as a ligand. A ligand is anything that is bound by a protein. These ligands bind to the protein by non-covalent bonds. In most cases, many ionic bonds are required to stabilize the interaction between the protein and its ligand. And the more similar the shape of the two entities, the more likely that they will form these non-covalent bonds between their atomic structures. So the specificity of a protein, be it an antibody or an enzyme or some type of structure within the cell, the specificity is defined by its surface, and that is known as its shape. Another physical feature that students need to be aware of is known as the affinity. The affinity can be defined as the strength of binding between two objects. So if you have a protein, such as an enzyme, to which two things can bind, and one of those things binds with greater affinity than the other, then the first thing is bound with greater strength. Affinity is based on the shape of the two objects, the charge distribution along their length, and the number of contact points that are close in terms of atomic distances between the two. This is illustrated in this figure, where the protein is represented by the green structure and its ligand is the yellow structure. So as you can see, the surface topology, i.e. the shape of both objects, is complementary to a large degree, and if they approach each other through random collisions in the correct orientation, they will then form a brief interaction based on weak non-covalent bonds. And the number of these bonds that are formed will determine the affinity by which the two objects are held together and the time duration over which. So to reiterate once more, the form and function of a protein are linked. That means that the anatomy, the shape, and the physiology, its function, are linked. All proteins have preferential sites along their surface to which ligands bind. There could be one binding site or more than one. The number of binding sites is dependent on the protein and its shape. Some proteins can change their shape to reveal alternative binding sites, while others do not. Another feature of ligands and proteins is that sometimes the binding of a ligand can control, i.e. regulate, the activity of the protein to which it is bound. And we'll see examples of this coming up very shortly in this chapter. Now, don't forget, please, that it is not just the amino acids along the surface of the protein that control its configuration. The interior amino acids also have a voice in which direction the polypeptide chain folds and how the protein's final shape is determined. So changes to amino acids, whether they're on the surface of the protein, at the binding site, or any other location, interior or exterior, may have an impact on the shape of the protein, therefore affecting its behavior. Antibodies, which are proteins, illustrate very nicely the properties that we just discussed. Our immune system is alerted to the presence of pathogens by detecting their proteins, which they release from their surfaces. The binding of the two types of protein is necessary for proper function of the immune system. The affinity by which the antibody molecule binds to the ligand, i.e. the protein of the pathogen, is a very important component in determining if and when the immune system is activated. The antibody molecule itself has peptide loops at one end which are compatible with the surface of the protein from the pathogen. These tips make the molecule have a particular shape. In general, an antibody molecule looks like a Y-shaped protein. It is composed of 2 light chains and 2 heavy chains. The 2 light chains are identical, and the 2 heavy chains too are identical. The antigen binding sites are located at the 2 tips of the Y. These have to be compatible with the surface of the bacterial protein. If the affinity of binding between these tips is of a certain threshold, then the antibody molecule will achieve its function. Another example is provided by the enzyme lysozyme. Lysozyme is present in tears and some other bodily fluids. It is a natural antibiotic because it has the ability to lyse bacterial cells by breaking the polysaccharide chains in the outer wall of bacteria. Here the enzyme is represented by blue, and the polysaccharide of the bacteria is represented by the red. The specificity of binding between the lysozyme enzyme and the polysaccharide ligand is high enough such that the enzyme will cut these chains repeatedly. Over the last few decades, with an increase in our understanding of how enzymes work, and with better and better tools for studying these interactions, biochemists have come to the conclusion that enzymes, in many cases, are able to change their own configuration during the chemical reaction such that they favor the production of the products. The first panel here reiterates what we already know, that an enzyme encourages the substrates to face in a chemically favorable direction so that a chemical reaction can take place. Panel B further tells us that the enzyme may force a rearrangement of some of the charge along its surface and the surface of the ligand, which may then also promote a chemical reaction. And then finally, some enzymes physically change their shape to cause the substrate molecule to be contorted in such a fashion that, again, it encourages the rearrangement of chemistry. Regardless of which combination of mechanisms the enzyme uses, it's important to realize that the shape of the enzyme is always restored to its original starting shape by the end of the reaction. Let's pause for 1 second to consider the role played by coenzymes and cofactors. Some proteins in nature can only adopt their active configuration with the aid of other chemistry. So in the case of blood, the heme group is necessary to bind with the globin protein to make hemoglobin. Only then can that molecule carry oxygen at the location of the iron. The same thing is seen in another protein that is found in the eye. Retinol is a light-sensitive pigment made of vitamin A, and it too has a partnership with a protein called opsin, which enables the eye then to interact with light energy and convert that to a molecular motion energy in the cytoplasm of these cells. We now move on to regulation of biochemical pathways. Whenever the student encounters the word regulation, they should substitute the word control in its place. So regulation and control are synonymous terms, and that substitution better helps students understand how the following section works. As we have tried to emphasize in this course, a cell itself doesn't have its own brain. Therefore, decisions have to be made by something, and that something turns out to be the biochemical pathways themselves. So as illustrated here, we have a biochemical pathway where a substrate B is converted to substrate X, and then Y, and then Z. In this biochemical pathway represented in yellow, an enzyme is present at each step. So there's an enzyme here at the red block, there'll be another enzyme between the X and the Y, and there'll be a third enzyme between the Y and the Z. In this particular example, all we are interested in is the interaction of Z with this enzyme here between B and X. As the concentration, i.e. the number of molecules of Z increases in the cell, they are more likely to bump into, by random motion, the enzyme in red. The enzyme in red will have a location on its surface which would then be able to bind temporarily the molecule Z. The binding of Z to the red enzyme will cause a change such that its active site for B no longer is available. Therefore, this pathway would automatically be downregulated or shut down. Downregulation simply means slowed down. So as you can see here, this is a self-contained logical pathway, and we call that a negative feedback pathway, in that there's a negative effect on the rate at which this pathway works as the concentration of the product Z builds up inside the cell. Now the opposite is also true. As the cell begins to utilize this substance for something else, Z, the concentration of Z inside the cell will begin to drop. There'll be fewer and fewer Z particles inside the cell cytoplasm, and the bound Z to the enzyme red will be more likely to release itself because of the ionic interactions and the kinetic energy, and restore the enzyme Z back to its normal configuration, thereby restarting the catalysis of B to X. So this self-contained system is able to regulate itself. Negative feedback systems are very common throughout the biochemistry of life. In reality, the biochemical pathways found inside cells are seldom found in isolation. As illustrated here, the interaction between one pathway and another can be quite complex. In fact, this is a very simple representation of how 5 different pathways can interact with each other. And every time you see a red line pointing back to an enzyme, that enzyme is under negative regulation by the product at the beginning of the line. So homoserine is a substance that's produced by this step in the pathway here, and homoserine will have a negative impact as its concentration increases, as we saw on the previous slide, on this enzyme, and this mechanism of negative feedback is very common. Let's now turn our attention to the enzyme itself in these biochemical pathways. Here we see a great example of how an enzyme can be inhibited by a product of its biochemical pathway. So this enzyme, which is part of the CTP biosynthesis pathway, CTP is a precursor to making more nucleotides. Cytosine triphosphate is indicated here as a red dot. So this enzyme is part of a pathway which generates CTP as its end product. The enzyme has 2 binding sites for its substrate. These are the active sites. And it also has 4 other binding sites, which are called allosteric sites. Allo means other, steric means shape. And these other sites on the enzyme can only bind CTP. So as the concentration of CTP increases inside this cell, because this pathway is on, the binding of CTP to this particular enzyme is increased, thereby inactivating this enzyme and therefore shutting down the pathway. So what you're actually seeing here is a molecular switch, and the switch can be in the on or off configuration depending on the concentration of CTP within the cytoplasm of the bacterial cell. Let us turn our attention now to positive feedback, which is different to negative feedback. In order to understand the biology of positive feedback, let's look at 3 very good examples. Let's start with example number 3. What this says is that students that study, they obtain better grades. The more they study, the better their grade. So what's happening here is, as one increases this variable, which is studying, the other variable, which is the grade, increases. Now naturally this cannot go on forever, because the highest grade that a student can obtain is an A+, or the amount of studying that can take place in a single day is limited to the number of hours in that day. Another example is provided by childbirth. Here the relationship is between the diameter of the birth canal and the degree to which the mother pushes on the baby. So as birth begins, the birth canal is quite narrow, and the amount of pushing that takes place by the mother is minimal. As birth continues, the diameter of the birth canal begins to increase, and the force by which the mother pushes on the baby also increases. This relationship continues until the child is delivered, when the system resets itself. And finally, the third example is provided by sodium gates on nerve cells. This example we'll see in Bio 110 and illustrated later on in this course. But let me give you a glimpse into how that works. As a nerve cell receives information from its cell body, it opens sodium gates on its membrane, allowing sodium ions to rush in. As the sodium ions rush in to the cytoplasm from the external environment, they open neighboring sodium channels. Those sodium channels also open and allow more sodium to enter the cytoplasm. This ripple effect continues around the cell membrane, and the number of sodium ion channels that are open increases, and the amount of sodium in the cytoplasm also increases until the nerve impulse reaches the far end of the axon. All three of these are great examples of positive feedback. This is perhaps the one slide in this chapter that may cause the most confusion. This is an example of positive regulation. As seen in the first panel, this particular protein can exist in two states: the inactive state with the open jaws and the active state with the clamped jaws. In the active state, this protein enzyme is able to hydrolyze sugar molecules at the beginning of a pathway that leads to the production of ATP, which is not shown on this slide. What is shown is the red molecule, which is ADP, adenosine diphosphate. Moving our attention to panel B, imagine that in a particular cell, the proportion of these enzyme proteins which are active is around 10%. Just by random molecular motion, some of these proteins would be in this configuration, whereas most of the other protein members would be in the inactive configuration. As the concentration of ADP builds up, because ATP is being used, the ADP molecules will tend then to bind to this enzyme, causing this molecule then to adopt the active configuration, which would lead to the hydrolysis of a large proportion of the sugar available, leading to the production of more ATP from the ADP molecules. And eventually, over time, the concentration of ADP would tend to decrease. They would therefore remove themselves from the allosteric site on this protein, and the situation would then tend to reverse back to panel B. The movement from panel B to panel C in the presence of ADP is an example of positive regulation, where the binding of a ligand to an allosteric site leads to the activation of a protein or enzyme. Had this slide been talking about negative regulation instead of positive regulation, the binding of a ligand to a target protein would have led to the inactivation of that protein. So that's the main difference between positive regulation and negative regulation. Once again, positive regulation results in the activation of a target molecule caused by a ligand, and negative regulation results in the inactivation of a molecule caused by its ligand. As we study more and more proteins and enzymes which are regulated in this manner, we discover certain patterns. One of the most important patterns discovered in the last few decades is the presence of different types of enzymes which modify proteins by binding to them different molecules. One such class of molecule is the phosphate group. Proteins can acquire a phosphate group from the environment, and they can also lose a phosphate group to the environment. Proteins which acquire phosphate groups from the environment do so in the presence of other proteins called kinases. So a kinase is an enzyme which transfers a phosphate from another molecule such as ATP onto the surface of a target protein. And in this analogy here, we could say that those enzymes, the kinases, they kiss and add phosphates to target molecules. And these are very common occurrences within eukaryotic cells. In order to remove phosphates from target proteins, the kinases are unable to do that reaction. So the cell has evolved alternative enzymes called phosphatases, and their job is just to remove the same phosphate group that was added by a kinase to that molecule. The amino acids of a protein which are commonly phosphorylated are generally serines, threonines, and tyrosines, STT. This slide shows such a phosphorylation event. In panel A, we can see that the light green target protein has an amino acid, a serine, whose side group sticks out into the environment, and under the right circumstances, a protein kinase will recognize the shape of this protein and phosphorylate it by leaving behind a phosphate group, and that is shown over here as yellow. As you know from a previous lecture, phosphate groups have two highly negative oxygen atoms, which then change the local environment to be very negative. These negatively charged oxygen atoms can therefore attract other side groups from neighboring amino acids or other molecules, thereby changing the configuration of this protein. The reversing process is to remove the phosphate from that particular serine and restore the protein back to its original state. We can classify proteins into two groups: those which are phosphorylated by the kinase and they are turned on as a result, and another class which are turned off by the phosphorylation event. Human cells are thought to have about 400 different types of kinases and a substantially fewer number of phosphatases. As you'll learn in another course, the main reason why many cancer cells become defective and grow out of control is because their kinases have become irregular. So a general principle emerges from studying proteins and their kinases. The activity of proteins within a cell can be regulated at many different levels. A cell may decide to remove a protein that is no longer needed by degrading it using various mechanisms. In this scenario, if that cell desires to use that protein again, it would have to go back to the DNA and synthesize messenger RNA, and that messenger RNA would then have to be translated by the ribosome into a fresh protein, pass through the Golgi, and then appear within the cytoplasm of the cell. A faster control mechanism is to have a molecular switch. A protein can be enabled and disabled, i.e. activated and deactivated, multiple times quite rapidly, sometimes many times per second. One remaining concept in this chapter, and perhaps the hardest to understand, is the case of GTP binding proteins. In general, these proteins bind to a small molecule of GTP in order to achieve some type of functionality. The bound GTP fits into a pocket somewhere on the surface of the protein. In this configuration, the protein is normally in an active state. The active protein then self-hydrolyzes its GTP, which means that it removes an inorganic phosphate from the surface of the GTP, and the GTP therefore becomes GDP. In this configuration, the entire protein becomes inactive. The only mechanism by which this protein can become reactivated is to replace the GDP molecule with a fresh GTP molecule. And this is done by disassociating the GDP from its surface and releasing that into the environment, and then acquiring a fresh GTP molecule which reactivates the protein to the on configuration. As you can see here, there's a slow step and a fast step. The slow step indicates that the disassociation of GDP from the surface of the protein can take some time. So basically, these proteins have built-in timers whose time can be varied by circumstances within the cell. So the slow step may be really slow in some circumstances, but the slow step may not be that slow in other circumstances, therefore influencing how long this protein is in the active state and how long this protein is in an inactive state. There are many such proteins within a cell, and they influence other proteins, such as those involved in regulating the cell cycle. A fantastic example of a GTP binding protein in action is provided by cells of the eye. In the retina, at the back of the eye, there is a layer of cells which detect light. They are referred to as rods and cones. They work in a very similar fashion. In the membrane of the rod or cone cells is embedded a protein which is sensitive to light. This protein interacts on the inside of the cytoplasm with another protein known as transducin. Transducin is a GTP binding protein. When activated, it does hydrolyze its bound GTP into GDP. That causes a conformational change in transducin, which leads to further changes within the cell. The time that transducin spends in an inactive state is influenced by many factors. Our understanding of proteins has led us to discover that there are many motor proteins traveling inside the cell along the cytoskeleton. These motor proteins perform a sequential change in their configuration, which leads to the protein actually walking along the cytoskeleton in a particular direction. This feat is achieved by having one of the steps and the sequential changes be dependent on a supply of energy, normally in the form of ATP or GTP. Therefore, it is very unlikely that the protein is able to move in the wrong direction, much like we do when we decide to move in a forward direction. The most sophisticated examples of protein interactions are provided by things known as protein machines. Protein machines represent an evolved complex relationship between a number of different proteins, both in their interactions and in the timing of those interactions. As indicated here, we have three different proteins which interact to perform a task for the sake of the cell. In a sequence of coordinated maneuvers involving many steps, some of which require energy, the proteins are able to achieve some fantastic outcomes. We will encounter protein machines in later chapters, especially with DNA replication. As our understanding of how proteins are regulated increases, some clear and fascinating outcomes have been realized. Proteins are further modified after their synthesis by ribosomes based on the cell's needs. For instance, protein X, shown here in dark green, can be covalently modified depending on the presence or absence of other signals within the cell at various residues along its length, influencing its behavior within that cell. As suggested here, there are mechanisms, some of which we're just beginning to discover, that permit the cell then to redirect that protein to an alternative outcome. Panel B gives us a real example of such modifications with a protein known as p53. p53 is very important in the cell cycle when the cell is trying to divide. It regulates the behavior of that cycle by determining whether and if different phases can proceed. So summarizing from the previous slide, we can use the word coded control. Protein functions can be adjusted and altered by the cell depending on circumstances. The final section of the chapter talks about protein studies, revealing how technology has enabled us to study and learn about protein function.